Hi everyone, so today we're going to be looking at monohybrid crosses. And so the study design key knowledge that you are required to know is the predicted genetic outcomes for a monohybrid cross and a monohybrid test cross. So I'm going to go through how to do both of those now. So before we begin, I just want to introduce you to some new terms. So when we look at different generations of organisms, whether that be animals or plants, right up the top we usually have the parental generation, which sometimes is indicated with a P. Then the first offspring is the first generation, often called F1, and then if we breed that generation, it produces a second generation, often called F2. So if you see things like P, F1, and F2, you know it's parents, first generation, second generation, and then we can even have F3, which would be third generation, and so on. So when we are doing crosses, we need to know the genotype of the parents. So here we have two parents. One has a dominant H, so the dominant allele codes for Huntington's disease, and then the lowercase h. So parent 1 is heterozygous, parent 2 is homozygous recessive. So two lowercase h's, two normal alleles, so they don't have Huntington's disease. So if these two parents were to have offspring, they can either, so this parent 1 can only pass on the capital H or the lowercase h. So if they pass on the capital H, they are passing on Huntington's disease alleles to their offspring, and parent 2 only has one type of allele to pass on, so no matter what, all of their gametes are going to have lowercase, and they're going to pass that on. This is, of course, one example of inheritance. Now, doing this way can get kind of messy. So the way that we can figure out what alleles will be passed on to offspring and what the genotypes and therefore phenotypes of offspring will be, we can use something called a monohybrid cross. So mono means one, and a monohybrid cross is used to predict the genetic outcomes of one trait. So in that example, we were looking at Huntington's disease. So that is one trait, and we want to know the genetic outcomes for the parent's possible offspring. So in this example, we have two parents with the capital H, and you can see here what their possible offspring are. I'm going to teach you how to do this monohybrid cross in just a second. We use this to predict the genotypes and phenotypes of offspring, and this square is called a Punnett square. And this can be used for both autosomal and sex-linked traits. So let's practice one. So this is an example of bobtails and cats. So bobtail is this short tail. It is an autosomal recessive trait, so it means it is found on an autosome, and the alleles that code for this bobtail is recessive. So here we have a male bobtail cat, and it is crossed or mated with a heterozygous female normal tail cat. So capital T is what we're going to use for a normal tail, like this over here, and lowercase t is what we're going to use for the bobtail. What I want to figure out is what are the possible offspring's genotypes and phenotypes in both a percentage and a ratio. So if these two cats had offspring, so they mated and they produced babies, what would their genotypes and phenotypes be? All right, so the first thing we would do is we would set up our Punnett square like this. So let me grab my pen. Okay, so the male cat up here, it has a bobtail, so we know its alleles are going to be lowercase t and lowercase t because it is an autosomal recessive trait. The female has a normal tail and is heterozygous. All right, we know it's heterozygous, so that means she has a capital T for allele for the normal tail and a lowercase t for her other allele because she's heterozygous. So the first thing we do then is we take this t over here and this t over here. We take the capital T, put it in the square, and then the lowercase t, because the mother is going to pass on this capital T while the father is going to pass on this lowercase t to their possible offspring. So this is one possible genotype if these two cats were to mate. So let's go through the rest and fill it in. So we've got capital T from the mother, lowercase t from the father, and now we're going to move over to the mother's other allele. So she has a lowercase t, and the father, of course, is going to pass on a lowercase t as well. And we'll do this again, so lowercase t from the mother and lowercase t from the father. So now we can see what the possible genotypes or the combination of alleles would be for any offspring. So any offspring that these two cats have would only have these possible allele combinations. They wouldn't have any others. Now I've just typed this up all neat here for you. So these were our options. So we know that over here, capital T and lowercase t, because this capital T is dominant and codes for a normal tail, this offspring will have a normal tail. Same with this one, will have a normal tail. These two down the bottom, however, will be bobtail cats. So if we're going to write the genotypes as a percentage, we know that one genotype is capital T, lowercase t, and that's 2 of the 4 are capital T and lowercase t, so that makes it 50%. And likewise, 2 of the 4 possible combinations are 2 lowercase t's, again 50%, and the ratio would be 1 to 1. Or you'd write 2 to 2 and then simplify to 1 to 1. So you go 2 to 2, and then you simplify. If you're going to look at the phenotypes, it would be similar. So we've got a normal tail for 2 of them. So 2 of the 4 will have a normal tail. Again, that's 50%. And another 2 will have bobtail. Again, 50%. So again, 2 to 2, and then you can simplify to 1 to 1. We're going to look at how to do a monohybrid test cross, which is a bit different to what we've just been doing. What we've been doing so far is looking at monohybrid crosses where we know the genotype of both parents. And so we're able to put that into a Punnett square and figure out the possible genetic outcomes for offspring. However, if we don't immediately know if an individual with a dominant phenotype is homozygous or heterozygous, we can do a test cross. So if one of the parents has a dominant phenotype, this could mean that they have 2 dominant alleles, or they could be heterozygous and have a dominant allele with a recessive allele. How we figure out which one they are is by doing a test cross. And so this is when we take that individual with the unknown genotype and we cross it with an individual that has a homozygous recessive genotype. So we know that they're homozygous recessive. Because they're homozygous recessive, we could assume that they have, well, we know that they have 2 recessive alleles. And so we're going to take this individual that is homozygous recessive and cross them with the individual whose genotype we don't know and figure out if they're dominant, homozygous dominant or heterozygous. If at least one homozygous recessive offspring is produced, we can assume that the parent is heterozygous. Because if we have at least one offspring, let me get rid of that. If we have at least one offspring that is homozygous recessive, we know that one of those alleles had to come from the parent, and so that means the parent with the unknown genotype is heterozygous. However, if only heterozygous offspring are produced, we can assume that the parent is dominant. So if all the offspring are producing that dominant phenotype, so that'd be like this, we can assume that probably the parent has 2 dominant alleles. This can get a little bit confusing, but we're going to go through an example now. We want to determine whether the genotype of this brown mouse here is homozygous dominant or heterozygous. So we know that brown fur color is dominant. So this guy we definitely know has that dominant allele. Right? White fur color is recessive. So because it's recessive, this mouse has to have 2 recessive alleles in order to be white. So we're going to do a cross of these two to determine, is this brown mouse homozygous dominant or homozygous recessive? And we're going to use the phenotypes of the offspring to figure it out. So let's look at this mouse here. So we're going to do a test cross. So we're going to cross this mouse with the white mouse. So I've put the genotypes in here. So this brown mouse definitely has a capital A. If they have a lowercase a, when we cross it with the mouse that is homozygous recessive, we're going to have these combinations, right? So capital A from the father, lowercase a from the mother, and so on. If it's heterozygous, this mouse, there is a chance that we will have at least one offspring that comes out homozygous recessive as well. However, if we cross the mouse and all the offspring have the dominant phenotype, so all the offspring are brown, we can assume that probably this brown mouse is homozygous dominant. That's because they don't have that recessive allele to pass on, therefore all offspring are going to be homozygous dominant. Though it's good to keep in mind that there is still a chance that every single time, even if they're heterozygous, the only offspring that are being produced because of the gamete combinations are brown. So there is a possibility, because we can't control which gametes are passed on, it could be possible that the brown mouse keeps passing on just their dominant allele, and therefore all the offspring produced will have the dominant phenotype. However, it's unlikely, if they're heterozygous, that they won't have at least one offspring with a recessive phenotype. So to recap, we wanted to know if this guy was heterozygous or homozygous dominant. We crossed it with a homozygous recessive mouse, so we know this mouse's phenotype and genotype, and we've got to look at the offspring. So if we have at least one offspring with a homozygous recessive phenotype and is white, we can assume that the brown mouse is heterozygous. However, if all the offspring are all brown, all have that dominant phenotype, we can say probably this brown mouse is homozygous dominant. So as usual, write down any questions that you have and bring them to class. We're going to go through a few more examples in class, so you get a lot of practice doing these Punnett squares.